species richness versus species abundance. Yes. So, when we look at these different habitats on the seafloor, like the abyssal plain, like cold speech, like hydrothermal vents, we're going to be using species richness and species abundance in order to compare these different environments and see kind of what's going on there. So, species richness is the number of different species that's present in an area. So in this classroom, we've got juniors and seniors. And so, let's say we have a species richness of 2, right? We have 2 different species, juniors and seniors. Species abundance is the number of each of the kinds of species. So in here we have, let's see, we've got like 4 or 5 juniors in here. No, more than that, like 7 or 8. And we got a sophomore too. Our species richness is 3, and our species abundance is going to be like 7 or 8 juniors, 1 sophomore, and then like 5 seniors. Okay, does that make sense?
1 5.
Okay. Approximately. So, species abundance is the number of each of the individual species. So, on the abyssal plain, your species richness is high, your species abundance is low. So you've got, in a given area, you've got several different kinds of species that you'll find, but there may only be 1 or 2 of those types. Okay, so that is the species richness and species abundance on the abyssal plain. We think that that's probably the case because the abyssal plain is so large that the animals are just spread out. Okay, so how do we actually sample the seafloor and figure out the species abundance, species richness, and what's going on down here? Well, there's several different methods that we can use. We have blind sampling methods. That's where we'll just kind of like drop air over the side of the boat and go to the bottom and then pull up whatever we find. So we don't really have any idea of what we're going to find. It's just whatever we pull up, that's what we get. So there's several different kinds of ways that you can do that, and I'll show you some of those. You can also use an ROV. The ROV is a remotely operated vehicle, so those are like the little robots that are attached to the boat, and then you've got an operator on the boat that's maneuvering that little robot. The main kind of like sampling that they do is with video. So ROVs are mostly just going to take pictures and videos, and then we'll see what's going on. You also have manned submersibles. You saw in the video Alvin. Alvin is a submersible that can make it all the way to the seafloor. So that can hold like 2 or 3 people inside of the submersible and then make it all the way down to the bottom. The problem with using manned submersibles to go to the bottom is it takes forever to sink down to those depths. So it could take them 7 hours to sink down to those depths. And then they get down there and they've got maybe 20 minutes to like sample or like set up whatever it is that they need to set up before they've got to start ascending back to the surface. So it takes a long time, it's expensive, it's hard to do. So we don't use it that often. Here's some pictures. So these are blind sampling methods. This one here on the left, that's an anchor dredge. So they'll dump this over the side all the way to the bottom and just kind of take a scoop out of what's on the bottom and pull it up. This is a dredge, or a sled. This will be pulled behind the boat, and then it'll kind of be pulled along the bottom. It'll stay on the top of whatever's on the bottom, and then anything that it comes in contact with gets shot into the back, into the net, and then gets pulled up. So that is a sled. Alright, here's some other types of sampling methods that we can use. We have grabbers and corers. So here's one type of like grabber. This is a box corer, so that's going to drop down, and then those are going to open up kind of like a jaws, and then they'll take a scoop out of the dirt. The gravity corer here, that's just going to sink down to the bottom. It's going to, because of gravity, stick into the mud at the bottom, and then they'll take and kind of like close off the top of the cylinder and bottom of the cylinder and raise up a core of the soil that's down there. And any animals that might be there. Here are ROVs. So this is the ROV Hercules, and then the bottom one is ROV Duncan at the Titanic. Okay, so they're down there looking at the Titanic. What's going on? And then over here, this is Alvin. That's our submersible.
Oh, is it the thing that they have in video?
Mm-hmm. That's the one that includes mine. Okay. So here's just some pictures of some things that we find on the seafloor. You saw the deep sea isopod. This is what it looks like in the water. Here's sea cucumber that we find down here, grenadier fish, and then another type of tripod fish. Alright. How we actually discovered that all of these life processes are occurring at such a slow rate on the seafloor was by accident. So they had an accident on a dive, and that's how they discovered that bacteria grow really slowly down here. So what happened was they were preparing to dive in Alvin, and so there was like 3 guys that were going to go on this dive down to the seafloor, and one of the guys had put his lunch in Alvin in preparation to go down for a few hours. And as you saw in the picture right here, Alvin is kind of like on this hook, yeah, that they'll use to like raise it out and then like actually lower it into the water, and then they'll release the hook and then it'll go into the water. Well, as the guys were like getting into the sub, the Alvin came off its hook. So they hadn't even closed the hatch yet. Like Alvin came off its hook too early, and the guys luckily got out, but Alvin sank all the way down to the seafloor, carrying the guy's lunch with it. And so Alvin's a really expensive sub. They, of course, wanted to recover it. So it takes a while to find another sub that can go that deep and get the resources and equipment that you need to recover that from those depths. So about a year later, they go back and they have everything that they need, and they go down, they bring Alvin up, and the guy's lunch still in there. His lunch was a bologna sandwich. So they pulled his lunch out of the sub, and the sandwich looks exactly the same as it did a year ago.
Whoa.
It hadn't decomposed at all.
Oh, the bacteria?
Because the bacteria grows so slowly down there. Whereas if you left a sandwich on your counter for even a month, it would be nasty, right?
Oh,
It would be
probably
pretty gross.
a day.
Yeah, it would be pretty gross. So the decay rate on like land happened much faster.
So wait,
That's-
doing the bomb there should be like meat whatever.
So that's how we discovered that the decay happens very slowly in the deep sea. We actually were at the time thinking about dumping all of our trash in the deep sea. Because, I mean, there's always been talk about, like, what do we do with our trash? Like, why waste land for landfills? Some people think we should send it into space. At the time, they were looking at sending it to the deep sea. But when we discovered that the decay rates happen so slowly down here, they were like, yeah, that'd be a bad idea, because all of our trash would never decompose. So we do not store trash in the deep sea, which is good.
Yeah, that's more of an idea.
This is a picture of the recovered Alvin sandwich. So that's what it looked like after a year. The only reason why it's got black dots is because it's an old picture, and so it's kind of pixelated. But it looks like normal. Maybe a little soggy, but normal.
Yeah, I took a bite out.
No. I think that's probably just like taking the sandwich apart. Alright. Another habitat that we find down here are deep sea corals. Deep sea corals. So these corals are going to be very different from your shallow water corals. We discovered them in the 1700s, but we didn't really know much about them until the late 2000s when we actually had the equipment available to go down and kind of study them. There's still a lot we don't know about them because it is hard to go down here. But here's some pictures of types of deep sea corals. Where we find them? We find them on the continental shelf and also on the tops of seamounts. So we find them between 250 and 6,000 meters. So very, very deep in the water. Compare that to shallow water corals. Shallow water corals must be in very shallow water because they need sunlight for the tubes belly to do photosynthesis. These guys are in very deep water. They're also in very cold water. So between 4 and 12 degrees Celsius is where they're found. Do you remember the average temperature that shallow water corals needed to be
20
in?
to 30.
20 degrees Celsius. It had to be greater than 20 degrees Celsius. So these guys live in cold, deep water. Shallow water corals live in shallow, warm water. The red dots on here, those are the pictures of where we found deep water corals. So So how they're different from shallow water corals other than where they're found. These guys do not have zooxanthellae because there's no sunlight down here, so zooxanthellae could not do photosynthesis, so there's no point to having them. So how do they get their food? Well, they will get their food from suspension feeding. The coral polyps are much larger in deep sea corals than they are in shallow water corals, so that they can capture marine snow and any sort of stuff that drifts by. So they will have very large polyps. They also grow much more slowly than the shallow water corals. So some of the reefs and corals that we found, we think may be thousands of years old because they are so large and they grow so slowly. Just like the shallow water corals, they are also going to provide a habitat. So we find a greater density of fish around areas that have deep water corals rather than in the areas that surround it. So they do tend to be a habitat. And other than that, there's still like a lot we don't know. So we don't know much about them still. We're learning. We do know that they are under threat though. So we think they're already under threat. And mostly because of deep sea fishing, commercial fishing. So when fishermen fish on the deep sea, they send these nets down, and these nets have these big like metal wheels on them that are used to, like, when the net gets drug over the bottom, that the wheels kind of roll and keep the net from getting snagged on things. So they pull these metal wheels along the bottom. But when you get like a giant metal wheel versus a coral, what do you think is going to win? The metal wheel. Okay, the metal wheel wins and crushes the coral. And there's been deep sea fishing in certain areas just for so many years that they've obliterated the corals in certain areas. So these pictures right here, they used to be deep sea coral reefs, and now they're just like crumbles of coral because of the deep sea commercial fishing. So you can see like the grooves from where like the net and the wheels have been dragged. Kind of crazy. I mean, like in one thing you kill 1,000-year-old coral, kind of sucks. So they are in danger. Our next habitat are cold seeps. Cold seeps. When we finish watching the video, you will see this. This looks like an underwater lake. So you're thousands of feet deep in the ocean, and it looks like there's a lake. It's crazy. They were discovered in 1984, and they're found away from the edges of the plates where you get methane gas that's bubbling out of the crust. So methane gas bubbles out, and you find cold seeps. Locations in U.S. waters where we find them: Gulf of Mexico, Monterey Bay, also off the coast of Washington and Alaska. Internationally, you find them in the Sea of Japan, off the Pacific coast of Costa Rica, and then off the Atlantic coast of Africa. So here, this picture shows you where we know there are cold seeps. So that's the purple dots. The yellow dots are where there used to be a cold seep. We found like fossils and stuff there, so there used to be a cold seep there, but there's not anymore. The food source for this area is different from anything else that we've seen thus far. So the food source for this area is actually chemosynthetic bacteria. Yeah, so these bacteria are able to take chemicals like methane or hydrogen sulfide and use the energy that's stored in the chemical bonds of the methane to make sugar. So photosynthesis uses the energy from the sun to power that reaction and make sugar. Chemosynthesis uses the energy from the chemical bonds and molecules to make sugar. So you can see the reactions here. So you have methane plus water is used to make glucose and then hydrogen gas. And then you've got carbon dioxide and water and hydrogen sulfide is used to make glucose and sulfuric acid. So they're able to use these chemicals to make sugar. So they are the basis of the food chain. They are the autotrophs. They're the primary producers. And they will support the whole food chain here at the cold seep locations. and then you can see the bottom of the photosynthesis reaction. So how that energy actually gets passed up the food chain, you will have bacteria that are doing chemosynthesis that are free living in the water, kind of like plankton. So they'll be moving around, and animals will be filter feeders or suspension feeders, and they will filter those animals or the bacteria out of the water. They can also have a symbiotic relationship with the bacteria. So worms and stuff will have a symbiotic relationship with the bacteria where the animal gets the glucose and then they give the bacteria what they need, like the methane gas. Or you can be a predator and eat things that eat the bacteria or rely on the bacteria. So that's how energy will get passed up the food chain. Cold seeps are going to be around for about 175 to 200 years before the methane gas kind of runs out and stops bubbling up. And then you will have, the animals will die because you no longer have a food source for them, so they will die. Okay, and then another thing will form, and the animals will move, and they'll colonize that cold seep, and then you'll get other animals growing and living. Okay, so here's what this looks like. This is a cold seep. So one of the chemical reactions that occurs actually creates like saltier water than seawater. And the seawater that becomes more salty will it'll become more dense, and so it fills in these depressions on the sea floor, and that's actually how you get what looks like an underwater lake. So you'll see in the video, it literally looks, it's so strange, because it looks like it's a lake underwater, and you're like, this is weird, because I am underwater, so how do you have a water, yeah. Alright, species richness versus species abundance here at Cold Seeps. So, species richness is going to be low. There's not going to be very many different kinds of animals, but the abundance is going to be high. So there will be lots of them. So there may only be 2 birds and clams, but there's going to be a lot of 2 birds and clams. So low species richness, high species abundance. So it's the opposite pattern of the abyssal plain. You can see pictures. So some of the animals that live here, you can see some of the clams and then some of the 2 worms. So here's some more pictures. Here's mussels that you find at Cold Seeps, and then here is the close-up of the 2 worms that you find. Hydrothermal vents. Hydrothermal vents were discovered in 1977 by a bunch of geologists. So it was weird. Back in the day, they didn't really have the ability to have a video that would go that deep, like a camera that could take video to go this deep. So they would take readings, temperature readings, and then take pictures simultaneously. So they were moving the ROV around in the deep sea and around this mid-ocean ridge, and they were looking at the readings, and at a certain point, the temperature went from being freezing, because the deep sea's cold, right, about 4 degrees Celsius, went from being freezing, and there was this huge temperature spike. So it was like something's going on here, because it got super, super hot. So they looked at where the temperature spike was, and then they looked at the picture that corresponded to that, and they discovered these hydrothermal vet communities, which were a brand new ecosystem that nobody had ever seen before, which is kind of crazy. They're found on the mid-ocean ridges. Do you remember what the mid-ocean ridges are?
Where new crusts form.
Yeah, where new crusts form. It's an underwater mountain range made of volcanoes, right, where seafloor spreading occurs. These things occur here. How they're formed. There's the picture of the known vent locations. How they're formed. So here's my little mid-ocean ridge. Water that's around here is under a lot of pressure, and there's cracks in the crust here because there's a lot of tectonic activity. So there's cracks in the mid-ocean ridge. So the water gets forced into these cracks, okay, and forced down into the crust of the earth. As it gets closer down here towards the mantle, what do you know about the mantle? It's hot. So that water, which is cold, now gets heated up, okay, and it's absorbing minerals because it's in the crust of the earth, gets heated up, becomes less dense, and now starts to rise back through the surface, towards the surface of the crust, and comes and emerges out through the mid-ocean ridge. So it emerges out. When that water comes out, it is super hot, it is full of minerals, and it's very acidic. The ocean water, cold, not a lot of minerals, and very, actually basic, slightly basic. So because of that, when those two water types meet, there's a chemical reaction, and you actually get a precipitation reaction, and all those minerals and stuff that are dissolved in the vent water come out of the water. That's what you saw in the video, that it looked like it was like smoking underwater.
Yeah.
That was a hydrothermal vent. Okay, and that the smoky look, that comes from all of those minerals coming out of the water. Okay, and that precipitation reaction. So those minerals fall out and form like the chimney that forms the hydrothermal vent. So here's your comparison between the two types of water. Ocean water is very cold. Hydrothermal vent water is about 732 degrees Fahrenheit. Do you know what temperature water boils at normally? 212. So this water should be way hot. Like this water should be gas. But because of the pressure of the deep sea, it remains liquid. So it's superheated water. And it looks like it's smoking, but it's actually not. Vent water is very acidic. Seawater is basic. Vent water has high metal content and mineral content, and also has a lot of hydrogen sulfide in it. And it also has no oxygen in it, whereas seawater will have some oxygen in it. So when the two meet, chemical reaction. So here's a picture to help you see. So here's the seawater getting forced into the crust. It's heated up by the mantle and then rises back through the mid-ocean ridge. The food source here is also chemosynthesis, except for it's going to be reliant on hydrogen sulfide and hydrothermal vents. And then the energy will get passed up the food chain the same way. So either you eat the free-living bacteria, you have a symbiotic relationship with the bacteria, or you have you're a predator of something that relies on the bacteria. So that's how energy gets passed up the food chain. One of the most interesting things that we found down there was this tube worm called the Vistria tube worm. It's so strange because we pulled it up and we looked at it, and this worm actually has no mouth or gut. Yeah, so all it has is an organ where it harbors these chemosynthetic bacteria. So it harbors the chemosynthetic bacteria, and it has gills and stuff, but it's going to use those gills to actually pull hydrogen sulfide and oxygen out of the water for the bacteria. So it'll use its gills to grab hydrogen sulfide for the bacteria, and then the bacteria will give food to the tube worm. These tube worms are huge. They can be like 10 feet long, a worm. Okay, and also strange, like they bleed red blood, so their blood is red. So the scientists tell stories of like when they like landed, like it was weird because like red blood came up all around like the portals of the sun because they bleed red blood. So it's strange. And because vents don't live or last for very long, they think that these Riftia tube worms may be some of the fastest growing animals on Earth. So the vent forms. You get water that starts coming up in one area, and then it gets colonized by these animals. We don't really have much of an idea of how that vent or the animals find their way to those vents. I think maybe like they get carried around by deep sea currents, but I'm not really sure. That vent will stay active for about 10 to 40 years, and then because of like the tectonic activity that's occurring here, because that is the area where seafloor spreading occurs, the plumbing that actually feeds that vent could get moved and the vent water could get cut off. When that happens, you get no more water, which means no more hydrogen sulfide, which means no more bacteria, which means no more animals. so all the animals die. And then the offspring find a new bed. Species richness versus species abundance here. The richness is going to be low, and the abundance is going to be high. So that's going to be the same pattern as the cold seeps, but the opposite pattern as the abyssal plain. So there may be like 2 worms and octopus and fish, but there's going to be a lot of worms and octopus and fish.